Namaste, I'm Preeti. Today I'm discussing a topic that is known as the biological nitrogen fixation. So this whole content which I'm discussing is taken from book NCERT. So in the biological nitrogen fixation, because we are talking that is the biological, so means that the living body involving in this. That's why it is called as biological nitrogen fixation. What happens actually, inorganic material get trapped into the living system. You see in the photosynthesis. So in the photosynthesis, inorganic material in the form of carbon dioxide and the water molecule get trapped into the system and ultimately produce that is the organic material, which is called as the carbohydrate in the form of sugar, in the form of glucose. Then those material further utilized by the plant to grow and in the development. So what happens, those plants, because they utilize the inorganic material to produce the food by themselves and for their benefit, that's why called as autotrophic plants, and those autotrophic plant are called as the producer, and the other organisms which take these producers for their growth and development, those organisms are called as consumer, and ultimately after the death of the plant and these consumers, they are decomposed by the decomposer, which can be bacteria, which can be fungi, and ultimately these inorganic material goes back into the atmosphere. So this is the way how the cycles operate, of the mineral cycles mainly. So because we are talking about the nitrogen fixation, so what happens actually that the nitrogen in the form of dinitrogen mainly present in the atmosphere. So in the atmosphere nearly 78%. So what happens that is how they are fixed, the nitrogen, because this is the unavailable form for the living organism. We cannot take the nitrogen directly, and the plant cannot consume nitrogen directly, but how they will consume? So this nitrogen got trapped into the system, because we are talking about the biological system, so we will discuss it later, the how the bacteria will help this plant to fix this nitrogen. So this nitrogen get trapped into the system and firstly converted into the ammonia. After the ammonia, it is converted into the second form, nitrous. This is the nitrite, and then nitrite is converted into nitrate. Nitrate is the absorbable or consumable form which is uptake by the plant, and then plant, in the plant body nitrate get entered, and after entering into the plant body, in the plant cell, then this nitrate is again converted into the nitrite, and then after nitrite, that it is further reduced into the ammonia, because at the physiological pH this ammonia is present at the protonated form, that's why it is NH4. So after that, what happens, plant consume it. Okay, so in the plant body, how the ammonia or this nitrate get be utilized by the plant. So it is converted into the ammonia in the form of nucleic acid, various biomolecules, and this is the nitrite. When this nitrite is converted into the amino acid by the plant body, this whole process utilized 16 ATP per nitrogen, because 2 nitrogen is here. There are 2 nitrogen molecule, which is there are the 2 nitrogen element which get trapped into this amino acid. So whole total, that is the 16 into 2 means 32 ATP. This is the, that's why this is the energy consuming process. So some because this whole process plant cannot do it alone. So what happens, there are some free living nitrogen fixing bacteria is present in the soil, and there are some symbiotic nitrogen fixing bacteria. What is the symbiotic? Symbiotic, where the two living body came in contact, interact with each other, and they give benefit to each other and then fix the nitrogen. So that's why called as the symbiotic nitrogen fixation. Here the free living, the cyanobacteria, that is called as blue-green algae, in which the anabina, nostoc and acetobacter is present. So because they fix the nitrogen, that's why called as the nitrogen fixer. And what conditions are required for nitrogen fixation? So anaerobic condition means oxygen is totally inhibited. Then because the nitrogen is fixed, so of course the nitrogen deficit condition means that the limited mineral element in the soil, that is nitrogen. So they require nitrogen for their growth and development, so they want to uptake it. That's why this biological nitrogen fixation is done by plant. So atmospheric nitrogen is fixed by three methods. That is the industrial nitrogen fixation, you will see in the field. That is the urea fertilizer. When it is thrown into the field, then it is converted into the nitrate form. And I already told you here that the nitrate form is most consumable form, which is uptake by the plant, and the ammonia. Only these two form, the ammonia and the nitrate, these are consumed by the plant, so it is converted into the nitrate. So what happens if it is given to the plant in excess amount? Actually this nitrate when goes into the plant, and we people consume those plant in which the nitrate amount is highly exceeding. So this nitrate when goes into our liver system, so it is converted into the nitrite, and nitrite toxicity induce some carcinogenic phenomenon, and there are some dangerous effect on our body. So limited amount of urea fertilizer is to be used in the field. So next is the biological nitrogen fixation. In the biological nitrogen fixation, we are talking here that the free living nitrogen fixation, here is the symbiotic nitrogen fixation. So these bacteria help in fixation of nitrogen by the plant. So the next one is the electrical nitrogen fixation. So in the electrical nitrogen fixation, the photochemical reaction. What happens? Photochemical reaction, where oxygen and hydrogen helps the fixation of nitrogen, and the ultimate product of this, that is HNO3, which is trapped by the living system, and then it is also converted into this, that is called as nitrate. So what happens in the nitrogen cycle? Because this whole tells about the nitrogen cycle. So in the biological nitrogen fixation, the dead organic material firstly decomposed, and after decomposition it is converted into the macromolecule, that is protein, and protein get converted into the amino acid, because the basic structure of the amino acid, you will see here, that is the ammonia. So ammonia will get liberate from the amino acid, and the bacteria which helps in the conversion of that ammonia from the amino acid, that is the bacillus vulgaris and bacillus mycoides. And this process from the decomposition to the ammonia conversion, that is called as ammonification. And those bacteria which helps in ammonification, this process, that those bacteria called as ammonifying bacteria. Now the next process is the nitrosification. What happens after this? That is the ammonia get converted into the nitrite, and the nitrite is converted into the nitrate. So there are the bacteria, because this process is called as nitrosification. So the bacteria which help in this, that is the nitrosomonas and the nitrosococcus. The next is when nitrite is converted into the this nitrate form, that is called as nitrification, and the bacteria that help in this, that is the nitrobacter. So this whole process means when this NH3 compound, means the ammonia, when trapped into the when get converted into this, that is called as nitrate. So this process is called as nitrification. And the process, the ammonification and the nitrification, they join together, and these are called as mineralization, because this process, this process by through this process the dead organic material that converted into the mineralization after this mineral, and this mineral is uptake by this plant, which helps this plant in their growth and development. So when this nitrate or ammonia or nitrite is present in excess amount in the soil, then this form is get back into the atmosphere. So this process is called as dinitrification, and those bacteria which help in dinitrification, these are called as the dinitrifying bacteria. So this is the nitrogen cycle, this is the how nitrogen fix, and this is the these are some examples of the bacteria which helps in nitrogen. What happens actually in the biological nitrogen fixation? Actually, plant root cell, that is the plant root cell, and in the plant root cell this is the root here, and what happens at here, because when we need something we communicate to each other. When the plant is under nitrogen deficit condition, so they will talk to the soil bacteria which is called as rhizobium bacteria. This is the rhizobium bacteria. So how they will talk to each other? It will release isoflavonoids, or it will release flavonoids. When it release these two compound is sensed by these bacteria, that is called as rhizobial strain. When they come in close contact to each other, the next step is the curling of root here. This is the curling of root here. So after curling of root here, this small pocket is formed, and in this small pocket these rhizobial strain will get trapped. So here these are some rhizobial strain will get trapped, and after when they will get trapped, we will talk about this particular site. So what happens? This is the rhizobial strain, and this is the curling wall of the root. So they will come in close contact to each other, and when they will come in contact to each other, this wall will get dissolved. When this wall will get dissolved, this rhizobium will get into the system. So they will enter into the root here. When they will get enter into the root here, they will form a continuous cell lines, and this is called as infection thread. This is called as infection thread. And when this infection thread reach here at the plasma membrane of the pericycle, okay, when this will reach here at the pericycle, again the plasma membrane will get dissolved. Then the infection thread come inside. Here are the cortical cell present. The cortical cells are present here. So when thread grow more and more in the other cell, okay. So what happened then after growth? We will talk about a particular cortical cell. This is the particular cortical cell, okay. Here the infection thread comes. So in the infection thread here, the release of the rhizobial bacteria. When the rhizobial bacteria will get released into the cortical cell, then it forms a structure which is called as bacteroid, and in the bacteroid this rhizobial strain will produce a molecule of heme, that is the leg hemoglobin, where the globin portion of this leg hemoglobin is is produced by the plant cell, while this heme group is produced by the bacteria. Okay, so bacteroid, after the rapid and huge growth, rapid and huge growth, this plant gave signal to the rhizobial strain that stop dividing. When they stop dividing, so they will form a spherical structure like this and this on the root, of the plant. Okay, so these are called as root nodules. Here the rhizobial strains are living, and this root nodule helps in fixation of nitrogen, But how? In the root nodule, in the rhizobial strain, one enzyme that is called as nitrogenase, which is oxygen sensitive enzyme. So when nitrogenase help in fixation, what happens? It will reduce 2 molecule of nitrogen into the 2 molecule of ammonia. So this enzyme is very important, that is called as nitrogenase. And this enzyme which is called as nitrogenase, which is produced by the rhizobial strain, it is the iron sulfur and the molybdenum iron sulfur complex. It is very sensitive to oxygen. So what happens when the ammonia is produced by these rhizobial strain at in the structure that is called as the cortical cell in the bacteroids, in the nodules. So after this, the fate of this ammonia is in the tropical plant. The form of nitrogen, when nitrogen is present in excess amount, because this ammonia is highly dangerous or highly toxic. So this in the tropical plant, this ammonia will converted into the urides, that is the allantoic acid or the allantoin. When required by the plant, then they will produce nitrate. Again, will make component of amino acid of the nucleic acid in the tropical plant. The second fate in the temperate plant. In the temperate plants, they will produce amides. What are amides? That are the 2 amino acid that is called as the glutamine and the asparagine. So what is these urides and the amides are the storable form of the nitrogen in the tropical and the temperate plants. So these are some questions, the type of questions which can come into the some competitive exams. So the stored form of nitrogen in the tropical plant, that is the urides. The stored form of nitrogen in the temperate plant, that is called as amides. The question can be, what amides are produced from the fixation of nitrogen? So these amides are produced, that is called as glutamine and asparagine. One more thing, after the nitrogen fixation, when these urides and amides are produced from the ammonia, from the nitrate in the plant cells, so this is called as nitrogen assimilation. nitrogen assimilation. Next is the first stable product of the nitrogen fixation, that is called as ammonia. Question can be the in which form ammonia is present at the physiological pH. At physiological pH, ammonia can be present in the protonated form, that is called as protonated form of ammonia. So the number of ATP consumed to produce an amino acid from the nitrate. So recall the concept, you will find it the 16 ATP per nitrogen. The next is process by which the ammonia is produced from the dead decaying matter, that is called as ammonification. Ammonification, process by which the conversion of ammonia to the nitrate, and when ammonia is get converted into the nitrate, the process is called as nitrification. And the next is the plant which make the symbiotic association with the rhizobial strain. Those plants called as the leguminous plant. Question can be which family make the symbiotic association with the rhizobial strain? Then you can see the Fabaceae family. The main enzyme which play in nitrogen fixation, which play important role in the nitrogen fixation, the name of enzyme is nitrogenase. So these are some questions which can be important for the competitive exam. Thank you.